Welcome to another episode of the Jobology podcast. I'm your host, Brandon, and today my guest is a cancer researcher. His name is Alex, and he's going to talk to us about how different types of cancer need different types of research, and how our genes play a specific role in how cancer affects us. So without further ado, Alex. Thank you for being with me today, Alex.
Thanks for having me.
I really appreciate it. The way we kind of like to start the podcast, or like to at least create a baseline for everybody so they know what we're talking about, is if I were to, run into you and be like, hey, what do you do for work? How would you answer that versus what's your official job title?
Yeah, okay. So what I do for work, I would say the broad focus of what I do currently is I develop immunoassays.
Okay.
For use in characterizing the expression of predictive biomarkers. I know that's a
lot
of stuff.
Wow. All right. You're going to have to, you're going to have to break it down a little bit for me there.
That's a lot to unpack.
There's a lot of words I've never heard before.
Exactly. Yeah. I guess just generally an immunoassay, just in general, that's my main focus on when I work on developing. It's using antibodies to detect the presence of a protein, basically.
Okay.
So we're using the same mechanics in our bodies where, you get sick and your antibody will find the very specific marker on a cell that's maybe for the bacteria or the virus, sure. Right? When we did, the COVID virus.
Sure.
That happened, right?
Yeah, we can say that. That's fine.
This has nothing to do with cancer. But what happened was everyone got that vaccine and it allowed you to produce this thing called a spike protein.
Okay.
On your normal cells, you take it in the RNA, and then they would produce the spike protein.
Sure.
And then your body would learn to recognize this spike protein,
Gotcha.
And then the actual COVID virus had that protein on it, and so the antibodies, they stick onto that protein, and then they know, hey, we need to attack this with our immune cells.
Okay.
Basically. So we use that same mechanism, but rather than attacking or trying to teach your body to do it, we're doing it totally outside of the body. It's not taking place inside anyone's body.
Interesting.
And we're taking basically preserved tumors, and we're using antibodies that are very specific to a protein that we're interested in, and we're probing that, and then we're basically able to visualize, look at it visually under a microscope and see every place where an antibody bound to the protein that we're interested in, and using that, we're able to tell, the expression of this particular protein that's associated maybe with a good prognosis for cancer or maybe a less prognosis. We're able to kind of characterize that expression, basically.
Okay. Wow.
If that makes sense.
It does-ish. I mean, there's still some words that I'm a little fuzzy on, but sure. Okay. Is that is science and that kind of something you've always been interested in? Or was it something that came about you just figured, there's a job opening, I'll take this job? Why get into it in the first place?
Good question. Well, I would say always interested in science in general, but I grew up watching TLC and Discovery Channel. I always had documentaries about dinosaurs and space, and so I when I was a kid, I wanted to be an archaeologist because I thought you're just going around digging for dinosaur bones.
Indiana Jones style?
Yeah,
okay.
Indiana Jones. Turns out that's not really what they do.
Not quite. Okay.
But I always had that science interest,
sure.
But I didn't really consider myself going into science. I was actually, when I started college, I was a music composition major. I was always been a musician and writing, doing music and stuff like that. And then I took a chemistry, an intro chemistry GE class. And I was the chemistry really jived with me. I was I like this. I know how it works. It's
I understand
very cool.
it because that's something that just went for me right over my head.
Okay, gotcha. Yeah.
This ain't gonna be me.
I really like it. Yeah, it's very mathematical and very predictable. And then just kind of going deeper down that hole, I majored in biochemistry and molecular biology in college, and so it's kind of just understanding how the biochemical systems in our bodies work.
Sure.
So it's a lot of protein enzyme interactions and things like that. And so from there, I got my degree, and I was well, I gotta put my degree in this work. Right?
Yeah.
So
gotta figure something for this.
Exactly.
Sure.
So I started sending my resume. I went to school in UC Santa Cruz, and then I stayed up in the Bay Area because Bay Area and Santa Cruz and San Diego are the two biggest biotech hubs.
Oh, interesting. All right.
Here on the West Coast for sure. Yeah.
Okay.
I mean I think Massachusetts is another big
Probably.
biotech hub, but San Diego and the Bay Area, just like that's where all the jobs are basically.
Huh. Okay.
So I stayed up there and I just kind of set my resume out like every day for like
Yeah.
a month. I would send 2 or 3 out, and then I finally got like a lab assistant job
Nice.
at this company. It's actually a really good company. They don't exist anymore, but it was a lab assistant for a histology lab.
Histology. What's that?
So histology is basically what I specialize
Okay.
in, and that's why I'm a researcher that specializes in histology and the immunohistochemistry assays that I work up. Those are part of the histology lab work.
All right.
So
Okay.
histology in general, just so you understand that, it's like you think about it as like, when you see the slides, maybe if you've done like a bio class, and you have like the little microscope slides that you look at.
Yeah, and you put that little like film on top of it, drop something. And,
Exactly.
Okay.
That's kind of what it is. It's so typically they'll be stained. Like it's if you just look at like a 1 cell thick slice of something, doesn't look like anything. Because
Yeah.
it's basically see through. And so what happens is probably by the time that you saw it, you were looking at something that was stained with these different things. And stains like the nuclei of the cells will be stained like a purplish color,
Gotcha.
and like the other parts of the cellular components will be stained different colors. So it lets you like visualize them.
Gotcha.
So that whole
The
process of like
mitochondria.
the
Powerhouse
mitochondria,
of the cell. I
exactly.
know that one.
Yeah.
It's about the extent of my knowledge there. Biology knowledge. Okay.
So that whole kind of field of like taking a tissue, maybe like someone's brain or something like that, and like preserving it, slicing it 1 cell thick, and then staining it so you can look at it. That is generally kind of what histology is. So
Okay.
I started out doing that,
Yeah.
and then kind of what I do now is just like a specialized
Okay.
thing of that.
Gotcha. And then the path, I mean, you kind of mentioned it already of like getting a degree. Is that if I wanted to go into this with no degree, I have no knowledge, can I apply to this and get the job? Or are they going to be like, you need a degree, you need some.
I don't necessarily. I think there's some. So okay, so there is a lot of histology work it's like a super in demand thing.
Okay.
Basically every hospital has kind of like a
Department for
a
it. or
department
something?
for it.
Okay.
And pretty much most biotech companies
Yeah.
also have some kind of histology needs.
Okay.
Basically.
Wow.
Anytime you're like, oh, we want to do our research or develop something,
Yeah.
and we need to test it on actual tissue. At that point, you got someone doing histology. Either you send it out to another lab or you have your own lab in-house, basically.
Okay. Gotcha.
So what was the question?
More just like path, like,
Oh,
do
the
you
path,
need
yeah.
degree? Do you,
Yeah.
is it something that I can just kind of start doing?
So I would say probably the easiest path is just have your bachelor's in some
So,
kind
like,
of
biology?
some kind of bio-y thing. And for that, you can pretty much just apply
Okay.
any application and probably start as like a lab assistant and then kind of work your way up a little
Sure.
bit. But there is a certification also. It's called the HTL certification, and it's really just like you have some amount of hands-on lab work that you do that gets verified that okay, they're in the lab and doing this, and you take like a test and you get a certification.
Okay.
You don't necessarily need your bachelor's. I think you can like at least have an associate's and then get your histotech license.
Gotcha.
And also be working in histology.
Oh, okay. Okay. So there's multiple routes to doing this. It's not just like, hey, you got to take these classes and that's it and do that.
Exactly.
Interesting.
I didn't even know what histology was when I started.
Okay.
I was like,
That's
what
something
is
good to
this?
know.
I just needed a job. And so,
Yeah.
and I would say you don't necessarily need, I mean, obviously if you're doing research like what I'm doing, you need to kind of know your science stuff.
Yeah.
But a lot of the more routine jobs where you're just kind of cranking out slides and cutting them, people have like good careers, families that they support, and they aren't super sciencey, and they just do that as their job, basically.
Okay. Okay. So there's more than, like we kind of said, there's more than one path, but there's also more than one, position you could do within this field of like, okay, you're not just like pigeonholed into one thing. You can kind of spread out from that. Interesting. Something that I find fascinating with all the jobs that I talk to, because I think a lot of people, myself included, a lot of times we have an idea in our head of like, oh, this job does this, or like their day must look like this, when in reality, I have no idea. Like, I don't know what the heck people do. So what does like an average day look like for you? Like, are you going into a lab? I mean, I'm assuming, I don't think you could do this from home. That would be pretty wild. But like, is it a lot of emails? Is it a lot of like hands-on work? Are you in a microscope all the time? Like, what does your average day kind of look like?
Yeah. Okay. So, I would say in general, histology work is pretty much in the lab for the most part.
assume, yeah.
I work for like a big pharmaceutical company, so I do more it's less cranking out stuff in the lab and more kind of designing and working across teams. So
it's like creative work too. Like you get to be creative. Like
yeah, in your own way. I wouldn't say it's
in like a sciencey way.
In a sciencey way,
All
yeah,
right.
definitely. In terms of like, having problems and trying to figure out ways to solve them, and there's not something you can look up. You know, for the first, lots of that.
Interesting. Okay.
But I would say my day starts. I go into the lab. You know, I don't work hourly, so I just kind of come and go as the work needs to get done. So
what time do you typically like try and get in by?
8-ish. I like to go in early. Most people don't. I'm like one of the first people at that whole site.
Yeah, everyone's like, well, if I can come and go, it's like, please, I'm gonna take my time.
I like getting around 8-ish, and you know, I have an office, so I'll go into the office first, and I'll check my emails and see if there's any correspondence like after I left the day before that I need to kind of get back to, and see if there's anything new that's going on, and check to see what my meetings are for the day. So we'll work on my lab specifically. There's like 3 or 4 of us.
Okay.
We do kind of different characterizations. I told you I do the immunohistochemistry where we're looking at the protein. We have another person that specializes in quantifying protein in a different way.
Okay.
Via like Western blots.
All right.
We won't get into that too much, but it's just another way of trying to see what kind of proteins are going on. And then we have someone else who specializes in like the RNA quantification, basically. Okay. So there's like a pretty what's the word I want to say?
Like diverse,
yeah. But there's a big the data that we're all producing complements each other.
Oh, gotcha.
So if I expect to see, if I do something and there's a lot of staining on this particular protein, if another guy in my lab does it via Western, he should see also a higher trend on that.
Gotcha.
And
it's like correlations between
correlation. That's the whole point. Gotcha. Yeah. So any one particular thing that I do on its own is almost worthless.
Gotcha.
But when you add all 3 of ours together,
it creates
and it correlates to each other. Then you're like, okay, so there's something here.
Gotcha.
Something here.
Oh, that could be fun. Like hit when you all hit on something, and then it's like, oh wait, we found something. We know something. We don't know exactly what maybe, but we'll figure that out.
And honestly, the biggest ones are actually when we don't all hit. When there's something weird, and it's like, why
like
is
anomalies?
That's the biggest thing, honestly. It's like, why is yours like this and mine's like this? And you kind of do some digging, and you that's usually when you learn a lot of stuff.
Oh, interesting. So you can I think something that's interesting that a lot of people maybe they always think like, oh, I'll just learn in my successes. But it's like sometimes quote unquote failures you can learn a lot more from and be like, oh, that's gotcha.
It's actually not how it works at all in pharmaceutical. It's pretty much all failures.
Okay.
And you go but the thing is when it didn't, not failure, we're going to
no, that's what I mean. Exactly. But yeah, quote unquote failures.
Exactly.
Whatever.
But it's, for example, we're working on like experimental drugs and we're trying to get them into like early human clinical trials.
Okay.
Yeah, right. So I would say if we worked on 11 new potential ones in a year, probably all 11 of them don't actually go.
Okay.
Clinical trials.
Yeah, that would make
maybe 10. Maybe you have one that goes. Maybe.
And
then maybe that one starts clinical trials and then it gets canceled shortly after. So, but what we do is all the work that we do, we document everything. We say, hey, maybe this didn't work, but here's everything. And years later, someone goes back and something happens. That's kind of how, what's the drug? Viagra, right?
Okay.
So Viagra was
it was like a heart thing, right?
It was exactly. It was a heart medication and it kind of was like, it wasn't really working that great. They weren't really bringing it to market.
And then everyone's like, hey, I also have this side effect with it that seems to, this happens. And then it was like, wait a second.
Yeah, that's an example of something where I think it was in clinical trials and they reported this thing. And so someone went back over the data years later. The drug, didn't end up going onwards. And they're looking at these side effects. Like, maybe I can do some research on this aspect of it. Yeah.
Okay. Interesting. So then when you're, you come in, you sit down, you check your emails. Do you then whisk away to the lab and you're just buried in a microscope for the rest of the day? Is it
exactly? But so then I'm usually in the lab pretty early because what I do is I'm setting up these runs basically. So the whole process of, I think I told you that we're probing these proteins with antibodies and trying to be able to visually see not only this is expressing a lot, but where it's expressing and stuff like that. So I have to run these experiments basically. And
that sounds fun.
In the past, you would have done it all. I would have been there by hand doing every single thing, and it would have taken like all
And
day and
recording
hours.
and everything. Yeah.
Exactly. Now we have robots to do everything.
Oh,
So,
interesting. Okay.
so typically the night before, if I know I'm going to do a run, I'll have my materials all set up and ready to go.
Nice.
And the first thing in the morning, I'll have the tissue sections, 1 cell thick, already cut onto the slides basically, and like dried and ready to go. And then I kind of get my reagents prepared and get my antibodies and start figuring out what it is I want to do. And then I kind of load them all on the machine. Bam, it takes like an hour, maybe hour and a half to get all that ready to go. And then the machine kind of does all the dispensing of reagents and washing and buffering and all that stuff.
That can probably lead to more, I mean, not maybe correct isn't the right word, but like more accurate, because it's not human error that's half all the time through messing up or something.
Exactly.
Okay.
So anytime you can remove the human element from something, we do. Because humans we always make mistakes, basically. Yeah, so anytime you can have a robot do a human's job, we always have the robot do it. Although robots kind of suck sometimes, but
it doesn't do what you exactly want it to.
Exactly, yeah. So it's usually about 3 or 4 hours for that thing to run. And so during the day, I'm doing other things. I'm preparing new samples. I'm going to meetings. I'm doing emails and stuff like that. I also just, in general, I think I told you about the histology, the kind of the tissue preparation side of it. Yep. So at my company, we used to have a whole other giant histology lab, and we would all do that stuff. And they've since laid like everyone off, so I'm the only one that does that now.
Like, so you can do it all now. Have fun with that.
So I have more of that.
Okay. All right.
Takes up more of my time.
Gotcha.
But I still get lots of opportunities to do my assay development, which is kind of what I'm more interested in.
What's your thing?
Okay. It's my specialty.
Nice. And then what time, if on an average day, again, it's sometimes tough to like qualify average days, but what time are you usually like, all right, I'm done for the day, I get to go back home?
I would say around 3-ish is when I'm like, I'm pretty much done.
Yeah, this is...
It's more just kind of seeing, is there anything else I need to stay around for? Answer emails, do some more like computer work.
Schedule a meeting for 4:30, and you're like, oh,
that happens a lot.
Schedule a meeting for 4:30.
4:30 meeting got canceled today, thankfully. But yeah.
Okay, so I mean, that's like a benefit that maybe not a lot of people would recognize or know of. Like, hey, I'm not bound to the time clock. Like, I can kind of work my schedule, work what I want to do.
Exactly.
Interesting.
And sometimes there's just times where I have to work on the weekend, because it's not often, but it's like, oh man, we really want to present this data at this panel, or we want to, the due date for our submission for XYZ is happening soon. It's like, I gotta work on the weekend.
Yeah,
which happens sometimes, but I would say most times it's
Monday to Friday.
Yeah, it's Monday through Friday, and it's I can leave an hour or two early if I need to. Sometimes I'll stay an hour or two later, but that's less common.
You mentioned previously like the other people that you work with, when you guys kind of work together. Is that like, is it a team? Is it a hierarchy? Like, are you the boss? Is there somebody else that's a boss, or are y'all just kind of at the same level?
Yeah, I would say we're definitely a team. We're all pretty much at the same level, although I think they might have slightly different levels. Sure. I don't know exactly.
Tiers.
Yeah. But yeah, we're all basically a team, and we don't exactly work necessarily together. Like, I have my own lab. Oh. No one else in there. I have like not a giant, but I have like a full histology lab.
Nice.
I'm the only person that's in there. The other people that are part of my team, they don't know how to do any of that stuff, so they don't really go into there.
And then they have their own.
Have their own thing, exactly. Okay. So we're all kind of working on the same projects, but not working collaboratively necessarily together like that. Yeah. But though we help each other out all the time, though. Sure. The people that are closest to me at work, so we definitely do help.
And then,
each
like,
other out.
do you have a boss you have to answer to?
I do.
Like,
Yeah.
For the hierarchy, how does that work?
So we kind of they got rid of a lot of the bosses in the last few years.
Yeah, realizing
6 months or so.
2 people are doing the same thing. One
exactly.
Gotta go. Yeah.
So our company, I want to say at the beginning of the year, had a big, 20% basically just gone. So my bosses were all gone. I have a new boss now, and he's remote.
Okay.
So he lives like on the East Coast. So we'll do like, he's actually over here like maybe every 3 weeks, maybe a month or so like that. So we'll meet in person then, but we do video conferences and stuff like that with them. But
you're kind of like autonomous almost a little
very
bit?
autonomous. I don't have anyone checking to see what specifically I'm doing, really.
Okay.
More just like,
Dang, that could be dangerous for me.
exactly, yeah.
For me, that could be dangerous.
But
I just sit in my office all day. Like,
exactly.
I did everything today.
You try and just like, yeah. So in an individual day, I could just do that, right? I can just sit there and not do anything.
Sure.
But overall, I'm like, I have to have this data. You know,
Yeah,
we want
you
to have
stuff to present. And stuff
exactly.
Like
We want to have
sure.
all our stuff together. So in any particular day, I don't need to do any specific thing. It's kind of how, whatever I want to do and however I want to plan it. But
that's cool.
But yeah. So and then eventually we're gonna get like, from what I'm aware of, we're gonna have like someone that's just like above us, just our lab, because this guy, he kind of manages lots of other people. He's pretty high up. He's like 2 or 3 rungs above.
Yeah. Okay. Gotcha.
So sometimes we're talking to him, he's just like, he doesn't really get exactly the specifics, you know.
Sure.
But I think eventually we'll get someone that's more directly right, my boss, and I think it'll be actually better and easier because it'll be someone that I can just
like a direct report.
Direct report, and they should be at least kind of hands-on in the lab, so they'll be able to know what I'm dealing with specifically and be able to help me out and stuff like that.
Nice. This can be something that you say pass, and we'll move on to the next thing. This could be something that you go into great detail on, whatever you feel like. However, I do like to at least ask the question of, is there like a pay range you can provide? I mean, I don't necessarily want to pin you down on, hey, this is how much you make, but is there like a range for your position so that people can kind of have an idea of like, oh, is this, you went to school for a while, you got a degree. I mean, you got to be getting compensated somewhat for that. Like,
yeah, I would say there is a pretty wide range. It's also very dependent on where you are. So I was in the Bay Area, and I was doing. I had eventually worked my way up at that same company from a lab assistant to like a supervisor on the shift. So yeah, definitely in the Bay Area, like 6 figures is pretty common-ish.
Yeah.
That's like kind of,
you have to have that. Though,
yeah,
like,
you kind of have to have it. Exactly.
Yeah.
I would say here in San Diego, the pay is definitely less just across the board. San Diego, just for whatever reason, everyone here says we can pay you less because it's San Diego. I guess, or something like that.
That's the selling point instead of the finance.
But yeah, I would say for what I do, what I'm doing specifically, it's in the 6-figure range,
nice.
Low 6-figure range.
Sure. And then it's like salary and like benefits. Per, all that stuff.
Full salary, benefits,
nice.
PTO, and all that
cool.
stuff. I would say though, histology in general, like if you're just like a traditional histotech is what we would call it. It's probably a little bit lower than that. It's more like, probably 60 to 90, and then maybe more like a supervisory would have in the 6-figure roles.
Gotcha.
But once you get more into like the research scientist side of things and less than just like the routine histotech role, it's like usually 6 figures. And,
gotcha. I mean, I'm sure it depends too, like what kind of company you're working for. If you're working in like
like I work
pharmaceuticals versus like a doctor's office, something like that. There's probably different.
Like I used to work doing the same thing that I'm doing now, basically, but I was working. So now I work at one of the largest pharmaceutical companies in the world. But before I worked at just like a really small, kind of not exactly a startup, but like kind of like a startup. I was making a lot less.
The mom and pop versus the conglomerate. Okay,
exactly. I went from there to this one, and I was like, woo, it's a lot more. But yeah,
nice. If somebody were coming up to you and said, "Hey, I'm starting my job as a in histology or as a researcher," do you have a piece of advice that you would give them, like starting out?
Yes, absolutely. So I would say most people don't. If they're starting out, they graduated college. I don't think most people know exactly what they're getting themselves into, and I had no idea. No one explained it to me. So I would say yes. So there's kind of 2 big buckets, I would say, for the biotech world.
Sure.
There's the clinical lab space, which is I'm getting patient samples. This is like someone went to the doctor and, hey, this is their...
I found this thing.
They found this thing. They found this tumor. They're sending their blood, whatever. They're sending it in to a lab. And then there's like kind of the research side of things, basically, where it's not dealing with an actual patient who's waiting on an outcome. And those 2 kinds of work are very... I mean, the lab work is pretty similar, but the environments are very different. For example, like a clinical lab is very highly regulated. You're going to be doing one thing and one thing specifically, and it's going to be like the chain of custody, and is very important, and documentation, and like exactly what happened at every single step is super important, because if something went wrong with that, and the patient got the wrong diagnosis as a result, it's gonna be like
it's technically somebody's, I mean, I guess both sides are people's lives, but this is like directly one person's life that you're dealing with.
Exactly. And so the clinical lab space, that's where there's going to be the most amount of demand for jobs just in general. So there's lots of people. An example, like a hospital would technically be a clinical lab.
Okay.
But there's lots of other labs that aren't hospitals that are still taking in patient samples and doing things with them. So in those spaces, it's going to have good pay and great benefits, but you're going to be doing a more narrow, like, here's what you're going to do, and you're going to do that. And then maybe if you go to school again, or if you get a different certification or a different license, hey, maybe I can move on to this or something.
But you're going to be a narrow focus.
Yeah, it can be narrow focused, and it's going to be maybe, not necessarily high volume. It depends on the thing, but you're going to be kind of doing the same thing in a very repeatable manner, so that
could be
results,
monotonous.
Not necessarily. I guess
monotonous might not be the right word, but you know what I mean. Same. Gotcha.
In a way that you're going to always be getting the same results and working on the same kinds of things. Versus, I would say the more researchy, or you can also consider it kind of startup-y space. You're going to get the opportunity to get exposed and get kind of thrown in and do lots of different things.
Yeah.
I would say, unless you're at the larger, big pharmaceutical companies, the pay is going to be a lot less also.
Sure.
But I would say anyone starting out is do both of them. Get an experience doing both.
Yeah.
So I started off in a large clinical lab. And so I got to see and it was like, we were getting like 800 samples a day, and we need to turn them over through our lab in like one day, basically. So get them in, and by the time we're done, like by the end of the day, all 800 samples are out of our lab and like into a different lab that's in that company. They're going to do stuff on further down.
I can't even get a podcast edited in a day. I can't even imagine doing that.
So it's a well-oiled machine. It's like done very well. It's like everything has a place and everything goes and fits in with each other, and it's just like very well-oiled and efficient, I would say. And then going to like a startup where you get a chance to do all this different stuff, but it's like all over the place. Who knows what's going on? Like, what is this? Where am I supposed to do it? So there's a lot of that,
Yeah.
But my experience, at least in the more startup-y research side of things, I got the opportunity to do way more stuff and get that experience that directly led to me getting the job that I have now. So it's like both of those experiences, and doing it at a clinical lab and getting that experience and working my way up through the things, and then just going to like a startup-y researchy lab and getting a chance to do all types of stuff that like no one really knows exactly what's going on. How should we do this? Okay, well, here's Alex. Just have him try to figure it out.
Yeah. So kind of like keep an open mind. You know what I mean? Like, don't say I'm only doing this. This is the only thing I want to do. Like, try everything, see what kind of works for you. Yeah,
because you're gonna get like, if you're working at a smaller lab like that, you're gonna get your hands in lots of different things. Yeah. But they're not always doing things the best. Sometimes they're doing things kind of backwards.
Oh,
in some ways.
Okay.
Like, I've definitely experienced that. Versus, the larger ones, you're not gonna get a chance to get exposed and get that experience to do maybe more interesting things. Yeah. But you're gonna see like the way like a well-oiled, well-operating machine works, basically.
Gotcha. Okay, that's cool. I mean, I think that's valuable for people to know. Is like, hey, be open, try different things, find your thing that you like, and then go for that kind of.
And also, I would just say in general, like a lot of the I would say, for example, like just practical jobs, like, because I didn't know like what is an actual like lab job that I'm doing. Yeah. It's going to be a lot of histotech work. It's going to be clinical laboratory scientists too. So that's like another thing where clinical laboratory scientist is kind of like the histotech side of things, but you're dealing with, like RNA, DNA, the wet labs we would consider it.
Ooh, okay.
So it's. And that one requires a bachelor's degree, and then you do like a 1-year program after your bachelor's degree.
Okay.
And those are super in demand. Every hospital, every biotech company, they need those as well. So yeah, I would say if you're first starting out, those are the two biggest buckets. Sure. You're probably gonna start off as a lab assistant, right? But just understanding like Do I want to go towards the CLS route, which is clinical laboratory scientist? That's going to be like another little program that you can get into.
Gotcha.
It takes about a year. It's not super hard to get into or do, but it's going to be like a little extra time and money to do something like that.
Okay.
Versus like the histology side of things, where there's going to be a lot more work doing that as well. They have its own thing called HTL, the Histotech license.
Yeah.
I would say those are the two biggest buckets. If anyone's like thinking, hey, I might want to get into research biotech stuff, look up those two things, because those are going to be like the two biggest buckets, and from there you can really specialize and do whatever you want.
Okay, gotcha. And I mean, that's good to know too, of like, hey, this is a field that's in demand. Like people, so it's not something where you're going to go to school for it and then never use it. Like if you go to school for it, you can find some way to use it.
Absolutely can.
That's
There's
awesome.
Tons of jobs and everybody's hiring.
That's
and then
awesome.
I would say lastly, just if you wanted to do like, we traditionally think of just like the master scientist, the guy who's like the researcher who's writing the papers. Yeah. Those guys are all PhDs. So if you want to do that,
you got some work to do.
Go get your PhD.
Okay. Interesting. Good to know.
You can absolutely have like a full career and like not be really limited. I mean, there's some limits, but not be super limited at all with just a bachelor's degree.
Okay. Well, this is the point of now the podcast where I have some questions that I think are a little more job specific than kind of the general outline that we go over there. But the first question I have is there anything that's like quantifiable that we have discovered in cancer research either recently or semi-recently, or something that like we can look at and go, wow, like that's an interesting discovery or leap or something?
So I would say that the general thing is that here's where cancer research is, at least at this point, is that we are aware that basically there is no one cause of cancer. There's numerous causes of cancer. And I don't mean like, oh, I drank this and now I have cancer. I mean like on the molecular level, like what is causing this specific cancer? What pathway in the body, what biochemical pathway is causing this cancer from person to person is going to be different.
Sure.
In some ways.
Okay.
There's some that are pretty common where you're like, okay, if it's like this, I know I can treat it this way. But what just medicine in general is there are more in-depth ways of looking on the molecular level of what's going on specific to your cancer, to your tumor specifically. And here is the best treatment based on your specificity. So a big thing is like whole transcriptome genomics, where basically big word, where it's looking at basically all the RNA in someone's biopsy. Maybe let's say you get a cancer biopsy, bam, it gets sent off to some lab or whatever like that, and they do like a whole transcriptome genomics. We're looking at RNA expressions of all these thousands of different cancer genes, basically. Okay. And you get all this data and you kind of run it through algorithms, and it kind of can tell you, it's not exactly there yet. I wouldn't say it's 100% in use in a clinical space yet, but it's getting there. It's getting adopted, and as more and more like physicians and oncologists are like comfortable with this.
Like it will give you the prognosis, or it'll give you like a path for
treatment. Path for treatment. Exactly. Gotcha. Yeah. So the you'd have like a pathologist and oncologist would like look at the tumor. They'd be able to tell like, okay, it's like this grade, we see these kinds of cells. And then you would do the molecular test to see exactly what pathways are driving this. Okay. And like, oh, if we see these specific combinations of genes at this level, we know that like this combined with this is going to be the best way for like. Gotcha. And what they'll measure is like, of all the patients, they'll do like a statistical analysis basically of all the patients that had your similar profile and they got this treatment, this percentage of them were cancer free in 5 years. Basically, is kind of the 5-year recurrence is kind of the standard that they go for. Okay. Perfect example of this is the drug called tamoxifen, which is it's a breast cancer. Like so, most common breast cancer is going to be is considered ER positive, which means it's estrogen receptor positive. Okay. And so what happens is, let's say someone gets breast cancer, they'll do a biopsy, they'll figure out if there is cancer, and then they'll probably take the mass out at some point. And then on that mass, they'll run these molecular tests. And what they're looking for is 3 common genes that are like the main drivers of breast cancer in women. And it's the estrogen receptor, the progesterone receptor, and HER2 receptor, basically. And based on the expression levels of those different combinations, they'll recommend different drugs basically.
Gotcha.
So
Okay.
tamoxifen, if you're ER positive and the rest are negative, basically what we consider. So low expression on the other ones,
Right.
high expression of this estrogen receptor. This drug called tamoxifen, you can use that, and it basically like no chemotherapy.
Wow.
A lot of times, no chemotherapy is
Sure.
needed. You can just take just this hormone drug, and it has a super high chance
Gotcha.
of not having your breast cancer come
See,
back.
and then that comes back to like saying like the three it's to me, and maybe like this is just the lay person idea is like all of them are like breast cancer is breast cancer. You know what I mean? Like I would assume like, oh, you get that, but maybe so like if I get it versus somebody else gets it, it could be completely different based on our genes and based on whatever's going on. Okay.
Exactly, yeah.
Interesting.
And you would
Wow.
treat them differently.
Gotcha. Okay. Wow, that's interesting to know. That's something that I would have I've never thought of. I just assume it's the same thing. Like, okay. Wow. Is there something specific that like cancer researchers do versus like Alzheimer's researchers or different diseases? Like, I mean, you kind of touched on it there, I think, of like, maybe it's more at a molecular level. I'm not sure.
No, I would say no, actually.
Okay.
I mean, you're dealing with different. I would say no. There's not a huge difference.
Okay.
Honestly, and
Gotcha.
the techniques and the approaches that you would take,
It's all similar. It's just
it's very similar.
focused on a different disease.
Focused
quote
on a different
unquote.
disease,
Okay.
different pathology. There'd be different genes driving it, but the way that you would attack it, go at it, is going to be pretty similar.
Okay.
Yeah.
Interesting. Nice. Well, again, that's something that I love doing about this. Is like I would have assumed like, oh, we got to do this. They do their own thing. This is different. But if it's all, hey, we get it on this, we try and look at it from a molecular level, and we move on from there.
Like, for example, I'll show you like what most people would do. Let's say if you're trying to come up with a drug that treats Alzheimer's and another person trying to come up with a drug that treats some kind of breast cancer. What most people are going to do is they're going to start growing those kinds of cells, right? So
Sure.
let's say Alzheimer's, it's like what, myeloid plaques in brain tissue, right? I think like that.
What the heck?
It's basically these
Don't
plaques.
ask me that
It's
stuff.
it's like these plaques in brain tissue.
I didn't know there was going to be a
basically.
test.
Sorry,
All right, I should have.
Sorry, yeah.
I should have studied better. Okay.
But basically, what you would do is both would start off just growing these kinds of cells,
Sure.
like in a petri dish. Basically, it's cells derived from. I assume that don't quote me on this one because I don't do Alzheimer's research,
fine.
but I assume that they would grow some sort of cells from a patient's brain
Sure.
that had Alzheimer's. They would start growing them. We do the same thing with cancer all the time. So we have different cancer cell lines that was initially isolated from a person years ago, and now we just grow them in dishes, basically. And typically, you'll start to understand how are these cells working, basically. What's specific to these cells do I think my drug's going to work on? A lot of times what you'll do also after that is you'll take those cells, those human cells that you grow, and you'll actually start growing them in mice, basically. Like
Okay.
mice that have no immune systems will be able to like take up human cells and start growing like a human tumor.
Interesting.
They might be able to, I know for sure they do this for sure for Alzheimer's. They'll give them Alzheimer's and start growing the myeloid plaques in their brains, basically. That same thing that causes Alzheimer's in humans. They'll cause that same thing in mice, and then they'll start treating them, basically. So you'll treat the mouse that you started giving Alzheimer's.
Gotcha.
You'll start treating it with your drug. We'll do the same thing with cancer. We'll start growing a certain kind of tumor in a mouse,
And then see
and
what
then
happens.
we'll start dosing it different doses on different days. We'll have different what's it called? Different mice will get different doses on different days and things like that. And then eventually you'll kill them, and then you'll take their brains out if it's Alzheimer's, and you'll see, okay, we gave it this drug. Did we see less reduction in the plaques?
Right.
Same thing with the tumors. Like, did the tumor get smaller? And then you look at it on a molecular level, see what's driving this. So it's very similar approach, I would assume.
Okay. I'm like sitting here debating if I want to ask this question because I just came up with it. And I feel like it sounds so ridiculously dumb. And I know there's like no dumb questions, but man, this one seems kind of dumb.
Just ask it.
Is there any danger to you doing this research? Like, not that you're gonna get cancer from, but like if you drop something the wrong way, or you gotta cut. Like, I don't know. Is there some kind of like that you could get contaminated from your research?
I would say my lab specifically not, except there's one very carcinogenic compound that I work with. It's called DAB. It's usually fine. Like your skin does a pretty good, is pretty good at protecting you.
Sure,
Basically,
filters it out. All right, okay.
But it kind of binds inside your DNA and will give you mutations and can give you cancer. So, you don't want to get it splashed in your eyes, any cuts.
Okay.
I don't. In the lab, we're always wearing lab coats, gloves, goggles, and everything like that. So. That's really the only thing that I have to worry about. It's just the tab.
Gotcha. So there is there is one potential. Okay,
but there's no chance that I could get cancer from working with cancer samples.
I figured no, but hey, I gotta ask these things because I think somebody else might be sitting there wondering the same thing. So
I think the main thing stopping from getting cancer is just the fact that I'm not working with like actual live tissue is a big thing. Like this has all been like preserved basically.
Gotcha.
And also, our immune systems, right? Like if you get, if you get like a transplant, right?
Sure.
You have to do all the blood typing. It has to be exactly specific, or your body will fight it off. It's like that. If you get someone else's cancer cells inside you somehow, your body will instantly just kill those cells.
Like, we don't need that.
Hey, this isn't us. That's the whole reason why cancer is so bad, because it'll grow. It's part of you, right? It's not a foreign thing.
Specific to the person.
It's part of you. So your body doesn't recognize that this mass that's killing you
isn't supposed to be there.
Because it looks just like you. But if someone else's cancer was inside you growing like that, your body would instantly go for it.
Man, that's crazy how like in tune our own bodies are when like half the time I don't even know what the heck's going on inside me. Interesting. All right. That's basically all I have for you. I do like to ask before we get to the last segment here, where I'll give you some time to talk about whatever you feel like. Is there anything you thought I was going to ask, or feel like I should have asked, or thought like, oh man, I was prepared for this and it never came up, or I don't know, something like that?
Let me think. Thought I asked
doesn't have to be.
Didn't ask. No, I think you did a pretty good job, man. Like,
appreciate it.
I didn't want to get I don't want to get too bogged down in the science and stuff that. So I think it's good because there's a lot more sciencey, specific-y things that I could go into. But
but I think people that are looking for that kind of will go and like gravitate towards that. Where just the average person, if they're like, oh, this is interesting, maybe I want to go into this, they want that more like surface level as opposed to like drilling down into like these are the exact things we're doing. And
exactly. I would say the only I wasn't really expecting you to ask it, but the only thing I didn't touch on that I think is important. It's just the biggest thing for doing science in general is just like having quantifiable data. Like that is your output. Like anytime you run an experiment, you think that's like mixing something in a beaker and it turns a different color and you go, Eureka. You don't care about that. All you care about is numbers on a spreadsheet somewhere. That is always your...
It's not movie scientists. It's real movie scientists versus real scientists.
Exactly. So data is the most important thing. And you generate as much of it as you can, and then you find systems for like parsing that data, because you'll get a lot of data and it means almost nothing until you have ways of like statistically analyzing it and bringing in complementary sets of data, and you're able to like build a picture basically about whatever experiment it is that you're doing.
Right. Okay.
So I would say, to people that are like think it's mixing beakers in a lab, it's a lot more numbers on a spreadsheet is what you care about the most.
Less beakers, more numbers.
Okay. Yeah, exactly.
Gotcha.
But that's good. And I would say another thing is a huge part of bio now. If I was going back to school, if I was talking to myself, 15 years ago, is just what we call bioinformatics, which is just all the data processing of all this bio data is a huge thing. So if ever you're interested, definitely consider going.
So like organization too. Gotta stay like keep yourself organized.
Yeah, not necessarily organized, but I would say like if you're interested in going into biology side, definitely consider going into like the bioinformatics side of things, where it's just taking big data and using it to like bringing it down to something that's like manageable and malleable, and like, oh, okay, I understand how this relates to this, basically.
All right, interesting. This has been fascinating for me because it's something that I know absolutely nothing about. So it's nice to like sit and hear somebody that's like, no, this is what we do, instead of, here
we do some of that too.
Yeah,
but
okay, all right, interesting.
It's a lot less of that.
Gotcha. Okay, and then the last part of the podcast here, I always like to leave for my guests to either, if they have something they want to promote, if they have a cause that they want to talk about, if they have something that they feel like the viewer needs to hear, here's your time.
So I would say has nothing to do with any charity or free time or anything like that. But I am a competitive Street Fighter player, and so I'm looking for anyone in San Diego that's looking to play some Street Fighter.
Okay.
Definitely hit me up because I don't like doing the online stuff. So if you're in San Diego and you're like, hey man, let's play some games, like definitely hit me up because I'm looking for people to play.
That's cool. Oh dang, that's a whole nother. We'll have to have Alex back and talk about how do you become a competitive Street Fighter.
The new one just came out. Like I was not playing for a long time, and the new one just came out like 2 weeks ago. So I've just been like, I'm going
grinding. Okay, nice, dude. Well, thank you for being my guest today, man. Thank you for sharing your story with us. I really appreciate it.
Glad to be here. Thank you.
Thank you for checking out another episode of the Jobology podcast, and thank you, Alex, for sharing your story with us. Before I let you guys go, a couple quick reminders. Please make sure you like, subscribe, and follow at Jobology Podcast on all major social media platforms, and comment what jobs you guys would like to see next. Like we say to end every episode, make good choices.